आइए अब हम बात करते हैं इलेक्ट्रोफोरेसिस प्रोसीजर के हवाले से। इलेक्ट्रोफोरेसिस को हम 3 से 4 स्टेप्स में डिवाइड कर सकते हैं, जो मेन स्टेप्स हैं। सबसे पहले जेल प्रिपरेशन, जेल कास्टिंग, सैंपल प्रिपरेशन, देन सैंपल लोडिंग एंड इलेक्ट्रोफोरेसिस। सबसे पहले हमें जो इलेक्ट्रोफोरेसिस बफर की ज़रूरत है, जिसमें हमने एग्रोज जेल प्रिपेयर करनी है। So before starting all that process, we should make sure that reasonable volume of this electrophoresis buffer is available to prepare the gel, as well as to add the buffer in the electrophoretic tank. जेल कास्टिंग के लिए हमारे पास प्लास्टिक ट्रेस अवेलेबल होते हैं जेल इलेक्ट्रोफोरेसिस अपरेटस के साथ। ये पिक्चर आपको एक जेल कास्टिंग ट्रे शो कर रही है, दिस वन। अगर आप देखें, इसके दो एंड्स जो हैं दे आर ओपन। दीज आर दोज एंड्स वेयर वी हैव टू अप्लाई द टेप, एज कैन बी सीन इन दिस पिक्चर, सो दैट वी कैन add the liquid molten agarose in it, so that it should be solidified in this tray. Agarose gel preparation के लिए, keeping in mind the size of DNA under study, the conformation of DNA under study, we decide what percentage is required. Then we decide how much volume is required, and for that purpose we need to have the electrophoresis buffer in which we are going to dissolve the agarose powder for further process. Weigh the correct amount of the agarose powder and then add it into the required volume of buffer, electrophoresis buffer. For example, if you want to plan 1% gel with the final volume of 50 milliliter, then we can use 0.5 grams of agarose dissolved in 50 milliliter of the buffer. It will give you final concentration of 1%. ये जो टेबल इस वक्त आप देख रहे हैं, इस टेबल से आप एक गाइडलाइन ले सकते हैं regarding the size of the DNA you are going to study and the required percentage of the agarose. In this table, you can see as the size of the DNA increases, the percentage of the agarose gel decreases. So once we have a glass flask or we have a glass bottle in which we have added the buffer and added the agarose powder in it, loosely cover it with some Kim wipes, or if this is a bottle, keep the lid or the cover of the bottle loose and place it in a microwave or in a water bath. The purpose of this step is to dissolve the agarose powder in the buffer so that it can be used for gel casting. Once this process is done, the agarose powder is dissolved in the buffer using some gloves, insulated gloves. Hold your glass bottle, take it out on the working bench and add ethidium bromide in it. Required volume of ethidium bromide is added so that we can avoid the staining step at the end of the electrophoresis. And the recommended concentration is 0.5 microgram per ml of the solution or of the gel is recommended. Then mix this liquid by swirling so that ethidium bromide is equally distributed in the liquid. In the meantime, we already have ready the gel casting tray along with the tapes on its both ends. Choose the required comb so that the wells can be given to the gel. Place them in the tray, and then in the next step, pour your gel in that tray. This gel will take about 30 to 45 minutes to become solidified, and once it is solidified, it can be used for electrophoresis. This 30 to 45 minutes time is sufficient enough for preparation of the sample. This is called the waiting period. This waiting period can be used to prepare the DNA sample, and as you know, जैसे हमने बात की थी कि DNA sample को हमें loading बफर के साथ मिक्स करना है, उसकी जितनी वॉल्यूम आपको चाहिए वो पहले से कैलकुलेट करके रखें. एट दैट टाइम सिंपली मिक्स दीज DNA sample with the loading buffer and use this incubation or use this waiting time efficiently. Once this waiting period is over, remove the tapes from the ends of the gel, or you can do it later on. At this stage, pour a little amount or volume of electrophoresis buffer on that tray, like if you see here in this picture. The purpose to pour the electrophoresis buffer here is to remove the comb. अगर आप किसी किस्म का लिक्विड पोर किए बगैर कॉम्ब निकालने की कोशिश करेंगे तो जेल ब्रेक होने के चांसेस हैं. या तो यहाँ पे थोड़ा सा वॉल्यूम बफर का यूज़ करके कॉम्ब निकाल लीजिए, और दूसरी सिचुएशन कुछ ऐसे होती है कि टेप्स रिमूव करें, इस जेल कास्टिंग ट्रे को जेल और कॉम्ब समेत जेल कास्टिंग चैम्बर में रखिए, which is filled up with the electrophoresis buffer, and then remove comb at that point. Once you remove the comb, the gel along with the gel casting tray is in the gel electrophoresis container. Now this is the time to load the sample. The sample can be loaded using the micropipette and the disposable filter tapes. It is always recommended that use the standards at the extreme ends of the gel. The well number 1 and the last well of the gel should carry your DNA markers, and all other wells in between should carry your DNA sample which you want to study. Now the stage is electrophoresis. Close the lid, apply the voltage as per calculations, that is 1 to 5 volts per centimeter, and this is the distance between the two anode and cathode. If everything is set up correctly and the leads are installed or are attached correctly, then after starting the current flow, you will see some bubbles coming out from anode and cathode. This is because of electrolysis. If you don't see any kind of bubbles coming out from these 2 points, it means the current is not flowing through the electrophoretic chamber and there is some error. Within few minutes, it will be visible that the color or the bromophenol blue is moving towards the opposite pole. Run this gel for the required time, and when you see that this colored bromophenol blue or xylene cyanol is at an appropriate distance, turn off the electric current, remove the plug, remove the lid so that there should not be any chance of electric current. Now this is complete process of electrophoresis. If you don't have ethidium bromide during the gel casting process, then you have to stain this gel using the solution of ethidium bromide for 30 to 40 minutes at room temperature.